I don't need to repeat the bullet 1, bullet 2. So to develop the finite HBV cure, so we believe it's likely we need to have a variety of combinations of novel compounds which could inhibit HBV replication, reduce antigen production, and enhance HBV-specific immune responses. As you can see from this graph, what we have in Roche's portfolio is we have both viral-targeted assets as well as immune enhancers. So we do have three clinical stage molecules. One is HBV LNA, another is CPAM class 1 molecule. So we do have another immune modulator, which is TLR7 agonist. Everybody is familiar with pegylated interferon, so I'm not going to repeat for that. So our thinking is by combination of different assets in-house or with outside partners, we would achieve HBV functional cure. So the RO7049-389 is an orally administered class 1 HBV CPAM. So what you can see is HBV core protein dimers, basically, in order to get into the functional nucleocapsids. So there are two different ways to interrupt this process. One is through the type 1, basically CPAM, another one is class 2 CPAM. So for the class 1 CPAM, which actually can trigger the abnormal core protein aggregates eventually being degraded. For the type 2, which actually can form empty nuclear capsids, so without a functional, basically viral particles. For our CPAM molecule, so what you can see here is a nanomolar compound in terms of EC50, and it all active against all the HBV genotypes. In addition, it has no cross-resistance with nucleoside or nucleotide analogs. TLR7, as Mala has introduced before, so it's a broad spectrum immune modulators. It can trigger the activation of a couple of transcriptional factors, and also it can trigger the differentiation of plasmacytoid dendritic cells. In addition, it can upregulate co-stimulatory molecules as well as trigger interferon production and other cytokines, ultimately activating T cells and NK cells. Interestingly, it also basically triggers the differentiation of B cells into plasma cells. This molecule is basically a double prodrug. What it actually it does is in the liver, it actually goes 2 steps of modification, then become an active form of this drug. In our phase 1 study so far, we have dosed more than 100 subjects. It was safe and tolerable, and which justifies it moves further into clinical development stage. In order to assess the combination therapy approach, what we did here is basically we use an AAV-HBV mouse model to evaluate how a combination approach can basically change the anti-HBV activities in terms of immune modulation as well as direct antiviral elements. So what we use this model is after 4 weeks of infection, you actually you can see a productive, persistent infection for more than 5 months, which give us enough window to assess our combination effect. Not only we can assess anti-HBV biomarkers as a typical DNA S antigen, E antigen, or the antibody, it can also help us basically to assess immune aspects of HBV-specific T, B cell ELISpot, as well as cytokines, as well as interferon-inducible genes. Other accessible elements we can basically do evaluate is body weight, serum, ALT, AST, as well as PK profiles. So the first graph I'm going to show you here on this slide is basically how the TLR7 oral drug treatment can change the basically adaptive immune profiling. So what you can see here is we give the animal 4 weeks of time to establish the chronic infection. After that, we give 4 weeks of oral treatment of our TLR7 agonist. What you can see here is basically in the germinal center, the activated B cells. You can see we have a significant increase of the B cells which are activated. Additionally, when we do the ELISpot in both B cell as well as the T cells got from the spleen, what we see is a significant increase of B cell and T cells specific for S antigen. Next study, we actually address the question, is this anti-HBV activity relies on functional adaptive immune response or it is? So what you can see is on the left panel, what we have is immune competent mice. So when we give the mice the oral drug, what you can see a drop of the DNA as well as the S antigen. What we did here is basically it's quite contrast when you use a SCID mice, which is T and B cell deficient. We don't see any movement in terms of HBV DNA as well as the S antigen. So this tells us basically the antiviral effect of this TLR7 agonist also relies on the functional adaptive immune component. In this panel, so basically what we are trying to show is a combination of CPAM plus TLR7 in terms of viral load suppression and S antigen modulation. So what you can see is in the DNA, the viral load, both TLR7 agonist and CPAM treatment can give a reduction of HBV DNA. However, it's a combination treatment actually give you a more dramatic and sustained effect. When we talk about S antigen, so what you can see, either CPAM treatment or the TLR7 agonist treatment can reduce the S antigen level. However, what you can see, it's a combination treatment of the both agents give us a synergistic effect as well as a sustained reduction of S antigen. So very interestingly, we only observed the S antibody production in the combination treatment groups. We did another experiment. We coupled our TLR7 agonist with entecavir. So what we found here is very interesting when you compare the combination of the TLR7 agonist with CPAM. When we treat the mouse with TLR7 agonist, you can see you have viral load reduction, and then you have the entecavir, so which also trigger the viral load reduction. A combination therapy didn't change the profile that much. In terms of the S antigen reduction, so what you can see, TLR7 give us a reduction in S antigen, but TLR7 in combination with entecavir does not trigger a dramatic synergistic effect when compared TLR7 agonist plus CPAM treatment. And we don't have any anti-HBs antibody production in this system. We also did the gene expression profile analysis. What you can see is a combination treatment give us very unique pattern of gene modulation. So when we did the pathway and also the gene enrichment analysis, we did find that there is a robust immune modulation elements present in the combination treatment. So further analysis of those components may help us to identify early treatment efficacy markers. So with that, I wrap up my talk. So we have two interesting compounds. So both are in clinical phase 1 stage. And with this translational biology data, so it further supports us. So we may have a very good clinical hypothesis to be tested in further clinical development. Thank you.